Hello everyone, welcome to my YouTube channel. This is my very first video and I'm really excited to share it with all of you. I hope you enjoy it and learn something new today. So let's get started. Today's topic is chapter 1 of microbiology, bacterial taxonomy. Don't worry if this topic sounds difficult. I will explain every concept in very simple language with easy examples so everyone can understand it. Let's understand the meaning of taxonomy. Taxonomy has three parts. The first is identification. It means finding out which bacterium we are studying. The second is nomenclature, which means giving the bacterium a scientific name. Scientific names are used all over the world so scientists from different countries can understand each other. And the third is classification. This means placing bacteria into groups based on similar characteristics. Let's think about a school library. Science books are placed together, history books are together, and mathematics books are together. This makes books easy to find. So taxonomy does the same thing for bacteria. The next is conventional approach. Before DNA technology was available, scientists relied only on observable characteristics. They first examined the shape of bacteria. There are three common shapes. Cocci means round, bacilli means rod shaped, and spirilla means spiral shaped. Then they performed gram staining. These tests divide bacteria into gram-positive and gram-negative bacteria. Next, they observed bacterial colonies on agar plates. Some colonies are smooth, some rough, some white, and some yellow. Finally, they performed biochemical tests. For example, can the bacterium break down sugar? Can it produce enzymes? Can it survive in oxygen? All these results together help identify the bacteria. Let's talk about molecular approach. Modern microbiology uses molecular methods because bacteria that look exactly alike may have completely different DNA. DNA is like the instruction manual of every living organism. Scientists study DNA to identify bacteria accurately. PCR is used to make millions of copies of bacterial DNA. Then DNA sequencing reads the order of DNA bases. Another important technique is 16S rRNA sequencing. This gene is present in almost every bacterium and changes very slowly during evolution. Because of this, it is considered one of the best methods for identifying bacteria. Now let's understand numerical taxonomy. The word numerical means based on number. Earlier, scientists identified bacteria by looking at only a few characteristics. But sometimes two bacteria look almost the same, making it difficult to decide whether they belong to the same group. To solve this problem, scientists developed numerical taxonomy. In this method, scientists compare many characteristics of bacteria, sometimes 50, 100 or even more. These characteristics include shape, size, gram staining, colony color, biochemical reactions, growth in different temperatures, and ability to use different nutrients. Each characteristic is given equal importance. No single characteristic is considered more important than another. A computer then compares all these characteristics and calculates the percentage of similarity between two bacteria. For example, imagine you have two students in your class. One student scores 90 in maths, 88 in science, 92 in English and 91 in biology. And another student scores 89 in maths, 90 in science, 91 in English and 90 in biology. Their overall performance is very similar. In the same way, if two bacteria share 95 out of 100 characteristics, scientists conclude that they are closely related and place them in the same group. This method is more reliable because computers calculate the similarity, reducing human error and personal bias. Now, let's learn phylogenetic analysis. This is one of the most advanced methods of bacterial classification. The main purpose of phylogenetic analysis is to understand how bacteria evolved over millions of years. Scientists compare the DNA sequences of bacteria, DNA contains the genetic information of every living organism. If two bacteria have very similar DNA sequences, it means they probably evolved from the same ancestor. Scientists use this information to draw a phylogenetic tree. A phylogenetic tree looks like a family tree. In a family tree, brothers and sisters are closely related because they have the same parents. Similarly, bacteria that are on nearby branches of the phylogenetic tree are closely related. Bacteria that are on distant branches are less related. A real life example is, imagine in your family, you and your brother have the same parents, so you are closely related. Your cousin is related to you but not as closely. A person from another family is much less related. The phylogenetic tree works exactly the same way for bacteria. Now, let's talk about Burgess Manual. Think of it as the dictionary or encyclopedia of bacteria. Whenever microbiologists discover an unknown bacterium, they compare its characteristics with the information in Burgess Manual. The manual contains scientific names, shape, gram reaction, habitat, chemical properties, classification. It is used in universities, hospitals, research laboratories, and pharmaceutical industries. For example, imagine you hear an English word that you don't know. What do? You open a dictionary. So similarly, when scientists find an unknown bacterium, they open Burgess Manual to identify it. You may wonder, why do we need taxonomy? Actually, taxonomy affects our daily lives. In hospitals, doctors must identify the exact bacterium causing an infection. For example, if a patient has typhoid, the doctor must confirm that it is caused by Salmonella typhi before choosing the right antibiotic. In food industry, useful bacteria are used to make yogurt, cheese, vinegar, and probiotics. Correct identification ensures food safety. In agriculture, some bacteria fix nitrogen in the soil. These bacteria improve soil fertility and help crops grow better. In environment, some bacteria break down oil spills, plastic waste, and sewage. These bacteria help clean polluted water and soil. And in research, scientists discover new bacteria and develop vaccines, antibiotics, and medicines using taxonomy. Let's quickly revise today's chapter. We learned that bacterial taxonomy is the science of identifying, naming, and classifying bacteria. We discussed two approaches: conventional approach based on physical and biochemical characteristics, and molecular approach based on DNA and RNA. Then we learn about numerical taxonomy, where computers compare many bacterial characteristics to calculate similarity. After that, we studied phylogenetic analysis, which shows how bacteria are related through evolution. Finally, we learned about Burgess Manual, the standard reference book used by microbiologists around the world. Understanding bacterial taxonomy is important because it helps doctors treat diseases, researchers discover new bacteria, industries produce safe food, and scientists protect the environment. So bacterial taxonomy is not just about naming bacteria, it helps us improve healthcare, agriculture, food safety, research, and environmental protection.